Good afternoon. It's now my pleasure to introduce Faraz Ali, CEO of Tenaya Therapeutics. The format for this session is roughly a 25-minute fireside chat. If anyone has questions during the session, feel free to type them into the question box and we'll try to ask them on your behalf. So Faraz, thank you for joining us today. Would you start off by giving us an overview of Tenaya and its platforms and how manufacturing fits into the broader strategy of the company?
Sure. Thanks. Thanks for the invitation. As always, we love this conference that you guys put together with a unique focus on cell and gene therapy manufacturing. I'm Faraz Ali, CEO of Tenaya. I have a few slides I'll present to help answer that question, especially for those who are learning about Tenaya for the first time. We are a public company, so I'll make some forward-looking statements. These are standard disclosures. For those who don't know us, Tenaya is a clinical stage, publicly traded gene therapy company. We are uniquely focused on heart disease and have a vision and mission to aim and transform and extend the lives of individuals fighting heart disease through discovery, development, and delivery of potentially curative therapies that target the underlying causes of heart disease. We have a pretty broad pipeline, wholly owned, and it's deep and diverse that tackles both rare and prevalent forms of heart disease. We have integrated a lot of capabilities, including manufacturing, that'll flash up in a bit. And we've just got a strong leadership team in cardiology, rare diseases, and gene therapy. We have three advanced programs in our pipeline, TN-401 and TN-201, or gene therapy programs. We have a cleared IND for TN-201, so we'll be dosing patients this year in Q3. And we'll be filing an IND for TN-401 in the second half this year. Both the drug product for both of those gene therapy programs will be produced by our own wholly owned facility, which I'll know we'll talk about today. And we have a very deep and growing pipeline of other gene therapy programs. This is our pipeline as it looks today. You see those two programs I mentioned at the top, TN-201 and 401, for the leading genetic causes of some cardiomyopathies. You see some large prevalence numbers there for both of those programs. You see a small molecule in the program. We have not internalized manufacturing for small molecules. We've focused manufacturing capabilities on gene therapy. So any comments I make today are purely about gene therapy capabilities. And then, as you've seen below, a deep, diverse pipeline of product candidates for both rare and prevalent forms of heart disease. I will make one point here as it pertains to manufacturing strategy. The fact that we're going after large orphans and the fact that we're going after some potentially prevalent forms of heart disease in the future. And so you see some of these programs go from rare to prevalent. That was one reason why we made a strategic investment very early on to invest in both non-GMP and GMP capabilities, because we knew that we were going to have to achieve some breakthroughs in order to improve the scalability of our products and the manufacturing and bring down the cost of goods to commercially viable prices. So that's one reason why I invested in manufacturing. We have three product platforms, so we're multi-modality in our drug discovery, and so a lot of what we do is gene therapy, but it's officially we do drug discovery in a modality agnostic way, and indeed that's why we have a small molecule in the clinic today. But three product platforms: gene therapy, cellular regeneration, and precision medicine. And in order to go, these each solve very different problems that have historically plagued the field of drug development for heart disease. And at the bottom, you see some five, what we call our core capabilities that we internalized and integrated pretty much from the get-go: disease models, capsid engineering, promoters and regulatory elements, drug delivery, and indeed manufacturing. And we think it's the interplay in particular of those four on the right that allows us to have drug products with a therapeutic index and a profile that we think are appropriate for patients and for eventual commercialization. And manufacturing is a big part of our story. So the fact that we do some capsid engineering work, in fact, at this point, we've screened more than 1 billion variants. That's another reason that we chose to invest in manufacturing. We were never going to be working on off-the-shelf parts. We've tinkered with both capsids as well as promoters, even in our early programs. And we've seen directly through our own work the impact that has on the product attributes. So having control over that in the form of manufacturing is partly one reason we made the investment, because we wanted to make the adjustments as we adopted not off-the-shelf components for our product candidates. We wanted to be able to make the adjustments on our manufacturing side, including upstream, downstream, and analytical development and quality control. So that was another driver of our early focus on manufacturing at Tenaya.
Great. So compared to when you last presented at this event, and maybe if you have other slides on this, you can keep going. Yeah. But when when you last were at this event, your GMP facility had not yet built out. It is now built out, and you have your IND clearance for your lead AAV gene therapy program. So can you tell us a little bit more about the way that you built out the facility?
Yeah, it's a good lead. And so overall, today we have about 50 people who are dedicated to both non-GMP and GMP capabilities. That includes upstream, downstream process development, analytical development, quality control. So one strategic choice that we made very early on, of course, in the company's history, back when I joined in 2018, was to first invest in non-GMP capabilities. So that's reflected here on the left. What we call our Manufacturing Technology Development Center is right here, co-located with research at our headquarters in the San Francisco Bay Area. We've internalized both HEK and SF9. We scale up to the 200-liter in non-GMP facility. This allows us to, this is basically everything from our vector core to, production for IND-enabling studies, GLP IND-enabling tox studies. We can produce all the material for, experiments at any scale, and also work on proprietary approaches, which I know we'll talk about later at the intersection of research and manufacturing. But as you said, we then built a GMP facility. We made a strategic choice to do this in 2019. We broke ground on this really in 2021 is actually when we 2020, sorry, and then we were completely operational by 2022. And you can see here on the right, we have manufacturing capacity about the 200-liter and the 1,000-liter scale. You can see the seamless transition going from left to right, from small scale all the way up to the highest scale. We have now successfully done several runs at the 1,000-liter scale. This number is already out of date. It was designed from the get-go to enable global drug development, so to meet the requirements for both the FDA and EMA. It's been designed with a modular approach, so we didn't overbuild all at once. We have a 100,000 square foot facility divided in half, and about half of it has been developed for initial GMP footprint, and then we have the capacity to expand and scale out and scale up logically as we anticipate certain milestones. So very flexible design, and it has been designed from the get-go to support not only clinical production now for TN-201, and other product candidates, but potentially for a facility from which we could do an initial commercial launch and file for a BLA. One thing I really love about it is that we put this GMP facility right here in the San Francisco Bay Area. So it's actually less than a 45-minute drive, depending on traffic, from the Manufacturing Technology Development Center, the non-GMP facility. So we have seamless transition of people and processes and know-how going from the left to the right. In fact, some of the people who develop the process go and help with the GMP runs. Similarly, the stuff that we learn from the GMP runs feeds back into the research we're doing on our early processes to continue to improve on the product attributes and yields and scalability. So this is sort of our overall footprint. We've learned a lot, and I know you'll get into that now, but we've learned a lot from making this investment and from growing up our own products from the left all the way to the right at the 1,000-liter scale. And frankly, the fact that we had such a smooth and successful IND clearance without any hiccups, I think it was partly attributed to the fact that we made this investment in GMP.
So on the topic of the IND clearance, obviously CMC issues have often been cited as a reason for IND clearance delays. You said you had a smooth experience. Can you talk a little bit more about what you've learned from the process of going through the IND filing?
Yeah, I mean, I would say even before you got to the part of IND filing, we learned a lot just from the process of growing up our own process from the left to the right, as I mentioned earlier. Even going through the first engineering runs and the GMP runs at the 1,000-liter scale, and noticing whatever differences that we saw in the process and product attributes and making adjustments. So just again, we've learned a lot just technically by having our own facility and being able to dynamically adjust to information as we went. Similar with the analytical assays and all the things that we knew that we would have to have in place to support our IND. So we learned a ton just in the process of getting to the point of IND. And then indeed, when we filed the IND, I think one thing that we've just got a great team and a great leader. Kye-Hong Kim is our Chief Technology Officer. He's a veteran of gene therapy. And there was a certain amount of anticipation of what the FDA was going to ask for, and going beyond even the guidelines. And so we had done some additional work just in case the FDA asked us for things that might have poked around the corner of the guidance and say, like, what's next, and anticipating the next phase of development, including pivotal studies. And I'm really glad that the team had that anticipation, because sure enough, during the back and forth inevitably that happens during your IND filing through clearance, I would say there was a good amount of back and forth on non-clinical and clinical, but particularly on manufacturing. And so anything that they asked of us and from us, or wanted to know our thoughts about the next stage of development, we were ready with that. We were ready with, in fact, not just ready with verbal responses, or sorry, written responses, but we were ready with data that sort of anticipated where they were going to go on some of the key, product attributes or process attributes. So it was, I would just say that the team showed really well in its anticipation of what the FDA was going to ask for. And I can, I guess, safely and confidently say there was nothing that they asked for, nothing that they wanted to see for which we were not prepared. And I do think that is directly reflective of the kind of team that we've built, and the fact that we went ahead and made this investment versus relying on the expertise of somebody else.
And so how do you plan to use this learning in your next program? In other words, to what extent can you repeat the same assays or even cross-reference for subsequent programs?
That's a great question. And the short answer is a lot. In fact, the fact that the TN-201 IND got cleared, we think actually gives us, both on the research side, but also on the manufacturing side, it certainly increases the probability of success that our TN-401 IND should go smoothly. It's same capsid, so that helps. Different gene and different gene cassette design, and those differences matter. And again, because we've internalized these capabilities, we know how those differences matter. But there's so much that we have learned, and in fact, to the extent that there were any questions, we can incorporate that into our filing for the TN-401 IND, and so anticipate the questions that we know that we got asked in TN-201 and bake that into the TN-401 IND. So we think that the probability of success in TN-401 has gone up in terms of the IND clearance. So there was there's both a lot of carryover from TN-201, certainly assays, certainly the non-compendial assays are the same, but even some of the compendial assays, there's a lot of learnings from TN-201 directly applicable to 401. And then, like I said, we already know some of what the FDA is going to ask. And the fact that this was so proximal, right? We just recently went through this with the FDA. There's unlikely to have been a lot of change in thinking at the FDA in just within a few months. So a lot of carryover, we think, of learnings into the TN-401 IND.
Great. So maybe let's dive into the manufacturing process itself a little bit. You have the slide up there with the 293 and the SF9 capabilities and the scaled, et cetera. Can you talk a little bit about the advantages that you saw for SF9 and what are the major differences from HEC-293s?
Well, first I'd like to say that there's always been, we all know that there's always been a little bit of a question mark in the minds of some, at least, about SF9 relative to HEK, but it's worth pointing out that 3 of the last 6 AAV gene therapy approvals were products produced by an SF9 process, including 2 out of the 3 last year. So right now, SF9 is punching above its weight in terms of approvals and regulatory validation. So I think that's helpful. I think there's a role for both of the manufacturing platforms, and the fact that we've internalized both is indicative of the fact that we think that there's value in both of those platforms. We, consistent with our philosophy of being very sort of a high attention to detail, we like to compare every single product and every single prioritized capsid from our capsid engineering efforts. We do head-to-head comparisons in both HEK SF9 to see, are there any differences between them? And once we're confident that there are none, then and we get to see the scalability in at least at early stages of our scale process. And as long as we're seeing what we expect to see, then so far we've prioritized SF9 recombinant baculovirus. I'd say the main advantage for us is scalability, right? I mentioned at the pipeline that some of our orphan indications even are large, like 100,000, more than 100,000 patients for TN-201, which is the lead gene therapy program, more than 70,000 patients for TN-401, which is the next one. That is, an order of magnitude compared to some of the orphan, ultra orphan indications that many have cut their teeth on in gene therapy. So for that, you need scalability. You need to be able to produce these more reliably. And we've done so, I think, partly because of the rapidity with which we went from 50 liter to 200 liter and eventually scaled up successfully to 1,000 liter. Given that we had just built this facility, I think was facilitated directly by the scalability attributes of the SF9 recombinant baculovirus compared to HEK. There are modest differences that we have seen in product attributes between the two platforms, none of which have translated to meaningful differences in vivo efficacy. As we've, again, we always do that head-to-head comparison. So for us right now, the advantages of scalability is one of the main reasons why we've adopted this. And if we're going to go to even larger indications in the future, we absolutely need the scalability of SF9. Having said that, we see value in both. And we're actually, and we may talk about this later, doing innovative work on both platforms to improve the attributes of both so that we maintain that flexibility into the future.
Great. So just to follow up on that, in terms of the scalability there, this is your current scale. Do you envision needing to scale above the 1,000 liter for commercial applications?
Yeah, we've certainly actively planning for that. Whether we need to or not, TBD, but we certainly want to lay the foundation now for that possibility of scaling up to 5,000 liter or beyond for a future launch. And in fact, the facility allows us to scale out, as I mentioned, but also to scale up. So we deliberately picked a space that would allow us to go to bioreactors as large as 5,000 liter. And SF9 gives us the ability to scale up there. And with a product like TN201, should we be successful in the clinic, it would be very beneficial to be able to scale up to 5,000 liter, both to produce the material as well and support a robust launch, but also to bring down cost of goods and to give us more pricing flexibility for a larger orphan. So yes, that is part of our plan. What obviously we're not operating at that level today, but the seeds as SF9 allows us to consider that for the future.
Great. And so in terms of the analytic methods, can you talk a little bit about the kind of analytic methods that you use to evaluate the purity, potency, and other quality attributes of the products that you're manufacturing?
I would say that there's nothing unique. I mean, there's a set of analytical tools that my guess is we and all of our peers are using. There's compendial assays that you need for every product and program, and then they're one specific to your product. There are some analytical methods we had to develop that are more specific to our program, obviously for both TN201 and 401, and in fact, for both of them, they may be a little bit different. There are some unique attributes to the fact that TN201 is a structural protein and that incorporates into the sarcomere, and that these patients already have a certain amount of abundant protein that's already there. So I would say the fact that it's structural and that most of the patients are heterozygous mean that we need to be able to have analytical methods that give information in a way that might be different than some of the more well-established gene therapies today that often involve secreted enzymes and often where there's zero protein to begin with and you're producing everything that's missing. That's about the extent to which I'll go to some detail. But otherwise, our methods are, I'd say, pretty comparable. We've got an experience to what others are doing. We've got an experienced team who know what they're doing, who've done this work for a long time. Yeah, I mean, we're doing all the things that you would expect us to do on potency, on purity. We look at, of course, empty to full capsids. That's something the FDA is focused on. We do look at partially. I think it's not just about empty and full, but also partials and how do you adjudicate on the partials. Scalability. We're looking at all the product attributes in quite a lot of detail.
Yeah, you mentioned the empty, full, and partial. Is there, obviously empty, full gets a lot of attention in conversation regarding purity of gene therapy manufacturing. Are there other things beyond, partial that people should be thinking about, or are those kind of the main issues?
I mean, that's one of them. I would say that it's potency and purity work closely together, right? I mean, and they're ultimately, you need to have a product that has the infectivity, and that infectivity can be affected by your manufacturing process. So we have infectivity assays like everybody else. Then, especially as you're working on novel capsids, you need to make sure that that infectivity is also matched with expression. So you want to make sure that the capsid you're working on is expressing robustly. And so potency ends up being, right, a combination of infectivity and expression. And we have multiple analytical methods for both, right? And so often you're triangulating between different methods that you've internalized to make sure that the information is relevant. Some methods are better for one thing, some things are better than the other. And so I would say, partly because of who we are culturally, and partly because our research and our manufacturing teams are so closely linked together, they're co-located in the lab, they're just separated by a wall. We're often finding that sometimes it's the research team that is bringing some great thinking on the right potency assay, using some proprietary work that I'm not at liberty to talk about, but there's the things that the research team is bringing over that might help with, for example, a potency assay on the manufacturing side. And vice versa, sometimes the manufacturing team are thinking of innovations that will improve the infectivity of the product and the expression of the product that will have relevance when we think about developing a research candidate for a new program. So that crosstalk between research and manufacturing, I think, is really, really critical. I think it's one of the things that makes us unique and sets us apart to some degree versus others, and certainly versus others who have chosen not to internalize manufacturing. Research helps manufacturing, manufacturing helps research.
Great. And so kind of building off of that, what kind of other innovations is Tenaya exploring that could further bolster the manufacturing, either with respect to scale, purity, et cetera?
Yeah, so at this upcoming American Society of Cell and Gene Therapy conference, we will have several posters over there, and some of them are intended to just show, we know what we're doing, whether it's looking at formulation and, formulation that is more specific, and we think for our desired use cases in heart disease, it may be, improvements in reduction of adventitious viruses. It may be improvements in, again, scalability and showing the data that confirms the scalability. So you'll see more on just showing kind of, we know what we're doing. That's the spirit of some of the assays. But then you also start seeing some things. Some of it will be at ASGCT. Some of it will be in future conferences that'll show us are pushing the boundaries. And just to give you a preview of that, HEK is a platform that we haven't even used in these first two programs, but the historical problem has been scalability and yields compared to, say, SF9. And we have some approaches that we think are novel, that show that we can improve the HEK performance relative to standard HEK 293, as well as relative to some of our peers. But on SF9, the scuttlebutt around SF9 has historically been, you got great scalability, but do you have the same potency as the HEK platform? And we think we understand those differences between potency of SF9 and HEK. Again, those were not, meaningful in an in vivo context for us when we did the head-to-head comparisons, but we think we have insights into how people have seen those differences and maybe potentially how to close those gaps. And so we're doing some work on that front as well to improve the potency of SF9 over and above what is possible today. So I think we're continuing to work on a surprisingly wide variety of things, all of which we think will improve manufacturing as well as our product attributes at the intersection of research and manufacturing. So more to come. We'll let the posters speak for themselves as they come out, but again, as I said, that's one of the things that sets us apart, is just that close connection between research and manufacturing. We have these biology meetings at our company where the whole company gets together to review sort of the latest and greatest data. And I think it's telling that whilst traditionally a lot of that is reserved for research work, the manufacturing team also shows up and listens to that work, and often they're the ones presenting in that forum. So it's truly a joint research and manufacturing effort.
Great. I see we've reached the end of our session, so I'd like to thank you again for participating and for the great overview.
Great, thank you. Enjoyed it as always.